All right, so the topic today, I'm going to talk a little bit about how the strategies that we use to monitor mostly deer and elk, but also we'll talk a little bit about moose populations in Washington. So, I don't want to go back. I don't know.
John?
Yeah, I don't know.
I can do it here?
Yeah.
Okay, so just as by means of background, as you might imagine, especially with hunted animals, we have a need to sort of assess abundance through time. And that doesn't actually exist just for hunted animals, but for hunted animals it's particularly important. And so the information needs that we have are usually defined by the management objectives. And those objectives are not just biological. Sometimes they're really the intersection of biological, social, and recreational objectives. Typically the kind of data we need is data that we can define a current condition state. But we're also interested in monitoring through time. So that implies the ability to sort of track trend. But obviously, we want the monitoring protocols to be scientifically credible. Some of the issues that we face that are sort of challenging is that, as you can imagine, across Washington State, we have deer, elk, and moose populations sort of widespread. That's a lot of populations, that's a lot of land area, and so trying to accomplish that sort of monitoring goal at those kinds of scales, one, really expensive. If you can definitely define something that you don't have enough resources to do, but also just the workload, the sort of sampling efficiency is a real challenge as well. That's why agencies like ours have historically relied pretty heavily on things like aerial surveys. Aerial surveys are a really efficient platform for sampling things like deer and elk populations. But they have a downside, and that downside is that you don't see all the animals, and particularly when you introduce things like cover, trees, things that obscure your ability to see an animal. You have to deal with the fact that even though you're flying around and you're counting animals, you're missing some typically unknown proportion of the animal. So you need to be able to address that. We know we have strong seasonality effects on the distribution of these animals. We have segregation issues, for example, gender segregation. In fact, that's one of the things that Lewis is going to be working on is looking at the kinds of things that affect and drive sexual segregation in elk. And then, of course, the detectability thing, which can vary across the landscape. Detectability is particularly problematic in some places and for some animals. And you can imagine out here, it's not as big a deal as it is in some place like Western Washington, where you've got lots of dense forested areas. And some species, for example, black-tailed deer that live on the west side, they're really very, very difficult to deal with in terms of a sampling scheme and getting at any kind of demographic estimates. Not all animals have equal detectability, even on the same landscape, but the reality is that some of the methods that have historically been available to do this kind of work, some of those assume that they do have the same detectability. So we'll talk a little bit about that more. And of course, you're always dealing with what do we measure, what are the actual metrics we're going to measure, and at what scale are we going to do that. And then the really interesting one is that if you deal with the public commonly assumes that we do censuses. And when I say censuses, that means we go out and we count everybody that's there, right? We account for everybody. And the public oftentimes doesn't quite grasp that we can't do that. And it's not because we're dumb. It's because, the environment just isn't suitable to that. And in fact, for some animals, when you're trying to census them, they're doing everything they can to be not censused, right? They're trying to evade you. So, decades ago, what the kind of data that most people had were really tallies or counts. So we go out in some way, often with an aerial survey, like I said, and we count animals and we have a tally. But a tally isn't a count, a tally and a count aren't really an estimate. So the difference between the two, what differentiates a count from an estimate, is some ability to deal with this detectability issue. There are not that many sort of fundamental approaches to dealing with that, and I've listed them here. There's a lot of variants to some of these that are out there, but most of them trace back to one of these. And I'm going to talk pretty much about the top three here mostly today. So sightability correction modeling, Mark-recapture methods, you might have heard that term, and for large animals like deer and elk, we use a sort of an innovation of Mark-recapture that we call Mark-resight. And there's some variants on this. Things like double observer methods, you might be familiar or read in the paper about using fecal DNA to do estimates. That really is a Mark-recapture kind of approach as well. And then distance sampling, and there are some variants on that too. And I'll sort of explain each of those a little bit more. And then not really quite the same family of stuff, but we also sometimes employ population models, particularly when direct estimation is not all that feasible. So sightability models kind of hit the forefront, hit the literature about 1987. The first one for elk was published in 1987 from data collected in Idaho, and so we've done a number of these. I've done this at least 3 times in 3 different places. So this is kind of the way it works. So basically, what we normally do is we hang collars on animals like that, and then we do a series of sighting trials. For example, with an aerial survey. So we're going to go out there. We're going to do a survey like we would normally do a survey, and we're going to count animal groups, and we're going to be watching for these collared animals. And when we find a group that has a collared animal, we're going to measure a series of covariates, things that we believe to be logically associated with our ability to see them or not see them. And the typical kinds of things that have that kind of logical relationship would be group size. You'd expect to see bigger groups easier than smaller groups. There could be some gender differences, canopy cover obviously that provides obstruction, that's going to be potentially influencing. Maybe activity, our animals when they're active more easily seen. Snow cover kind of affects your sighting environment. Could be vegetation class, sometimes it's lighting. So these are the kinds of things that when you first go out and start collecting the data, you collect those kinds of data. So what you do is you go across your sampling area and you account for every collared animal that you've seen and you collect those data. Well, then what you do is you use the radio collars to go back and track down the ones that were there that you missed. And you collect those same series of covariates. So essentially you've got data now. Every animal in your data set had a 100% chance of being detected. You either saw it or you tracked it down later, right? And so everybody contributes data. It's not biased towards just the ones you saw. Then we fit a series of what are called logistic regression models, and if you're not familiar with what that looks like, that's a logistic regression model. It's basically a regression function that's constrained so that the outcome variable ranges between 0 and 1, so it mimics kind of a probability fairly well. So what we do is we fit a series of models where we have varying combinations of these covariates. Some would only have one, some would have all of them, perhaps. And then we use a statistical procedure to try to figure out from the data how does the data best fit with those models. So there'll be some that it doesn't fit very well with, there'll be some others that it fits better with, and we can actually do this model selection procedure and figure out, okay, among the models we've looked at, which are the ones that really best are supported by the data that we observed. It yields what we call a Horvitz-Thompson-like estimator, which is simply an estimator that accounts for inclusion probability, which is kind of what the problem is, right? So we're going to see some animals, and different animals in different groups have some sort of different likelihood of being seen. So that's what we're after. And so we ended up with this model. And here's these are just some citations for, this has been done a number of times for elk. I've got a couple on here. The original one, 1987, was here in the Journal of Wildlife Management. And so this is kind of what the data would look like, the actual observational data. So this is a real data set, 159 sighting trials, and as you can see, total groups, group seen, percent seen. So when the canopy the environment was very open, we didn't really have trouble seeing the animals. We saw virtually all of them, but as the canopy got increasingly more dense, that drops off, so that by the time you get up to, canopy cover is more than 60%, we're looking at less than 10% chance of seeing those animals. Right? Snow sometimes doesn't end up being all that informative, mostly because it kind of depends on the conditions under which your sighting trials are done. Group size, fairly predictable again. The smaller groups were missed more often than the larger groups. Gender effects, so cow-calf groups, these were winter surveys, so these animals are sexually segregated in the winter, and the bull groups are missed a lot more than the cow groups were. Activity, again, animals that were active had a really high chance of being seen. Ones that were bedded down and inactive were less likely to be seen. Across cover types, the cover types that were more open, like fields and clear cuts, pretty high rate of seeing them as opposed to missing them. When you get into the conifer stands, pretty high proportion are in the missed category. So that's what the actual data look like that you model on the basis of.
Scott?
Yeah.
How do you know the ones that you didn't see?
How
do you know what your actual interest was?
So we have telemetry, an antenna on the helicopter. We basically track them down, and so most of the environmental covariates are still pretty easy to get. I mean, we can track down. We know right when we're over the animal, so we can look and get the canopy cover, and we can get the other things. The group size is more challenging, but a lot of times this is what we're able to do is use the helicopter, again, monitoring the signal. We're able to, it's not a homogeneous place usually where they're at. So we try to move them around a little bit so we can get them to where we can actually see them, and then we make the count and collect those other kind of things. So it adds a challenge, but it's generally overcomable. So this is some work we did at Mount St. Helens. We had 331 collared elk trials, and the covariates, kind of the same list I showed you before that we looked at, and we generated a model. Really, the only two covariates that were really important and really pretty much told the story about who we saw and who we didn't were group size and percent canopy, and this is actually the logistic regression model, the linear part of it that we generated. And so we used that with the data. So these are actual estimates over a period from 2009 to 2013. This was estimates specific to just cow groups. And so we did 2 trials a year. And this was a time when we were actually, the harvest strategy, we felt we had too many elk in this particular area, and our hunting season structure was designed to sort of push the density down. And this is the estimates that we got over time. So you can see how it's reflected in what's happening with survival. This is total elk abundance in that same project, but for a different landscape scale. This is just for one specific game management unit, and again, you can see how the estimates played out from the Sightability model. So if you look again, there's been quite a few models published in the literature. So this is essentially a graph that shows for a fixed group size of 2, what's the effective canopy cover that is predicted by these various models. So this is that original 1987 Idaho model. This was one I did for my PhD. This is one that the Puyallup tribe generated, and Mount St. Helens and Nooksack, which is in northwest Washington. So you can see the various curves that show the effective canopy. And so one of the things you'll see is that this Nooksack area, which is that's a really wet environment, northwest Washington. You don't only have trees, but you can have multi-layered canopies, and wet series conifers. And what it shows you is that the effective canopy closure is really dramatic there. I think the sighting probabilities drop off a lot more rapidly there than they did in some of these other places that are more typical of sort of east side dry forest conditions where you have trees, but they don't have quite as strong of an effect. For a group size of 10, same thing, so the canopy closure. So you can see that on here, by the time you get to 60% canopy, in most of these models, your predicted sighting probabilities are pretty low. They're going to be 30% or lower, even though they're really high when it's open. Larger group size, so should have some effect, and so when we just look at 60% canopy closure here, but with the same model, you're getting predicted sightabilities that across some of these models are quite a bit higher. So in the Idaho model at 60% canopy closure, it's still predicting that you're seeing almost 90% of the animals that are there. So that's, these variables kind of interact and they influence the output. So here's another application we did for Columbia Basin mule deer. This was a situation where we had it's a pretty large area. It's not even the same in our organization. It's not even the same people that are monitoring deer across that part of the landscape. But what we had going on was most everybody was kind of doing their own thing, and they might potentially be, because of movement patterns of deer, they might potentially be counting the same deer. One guy's counting them here. Half those deer are getting counted a month later by somebody else who's doing their survey different in a different time frame. And it became pretty apparent that wasn't a very good strategy. And so we undertook this effort to really kind of integrate everybody and try to do a really collective, integrated approach. So we developed a sampling scheme. We used an existing sightability model for deer, for mule deer, and we were after abundance, age ratios, and sex ratios. It was a pretty large-scale effort, really. We used a stratified random sample design, so we basically stratified the sampling units that we had into units that we expected to have lots of deer, some that we expected to have sort of moderate numbers of deer, and some that we knew weren't likely to have all that many deer. That information did not come from the survey because that would be sort of taboo. So we used other kinds of information to pre-stratify. Then we did our surveys, used a sightability correction model. The area we were looking at was in excess of 4,000 square kilometers. And the way we sampled that is we couldn't sample the whole thing. It was too big an area. So we sampled 70% of the high-density units where we expected most of the deer in our count to come from. About a third or so of the medium-density units and a slightly less effort in the low-density units. And that's what the results look like. So the actual deer observed over 3 years across all those stratas, somewhere around 5,000 deer. So then when we run it through the sightability model, deals with both the detectability, but it also deals with the fact that you sampled, you didn't count all the units. And so the estimates at the end of the day for those 3 years where we counted about 5,000 deer, the estimates are actually in the vicinity of 12 to 13,000, a little bit more. If you look at the components of the variance, so how much of the sort of anticipated error or predicted error are associated with those estimates, you'll see right away that the biggest source of error was the fact that we didn't count all the units. The sampling error itself, so the variation across the units when predicted, when you basically take the low density units you flew and try to use that to predict what was in the low density units you didn't fly. So the variability within the ones you did ends up really inflating the estimates. So what that tells you, if you want better estimates, in this case, what's the goal? Where should your effort go? Fly more area, fly more of your units. The variance associated with the actual model error were pretty small, as was the error that's associated just with the fact that you didn't see all the animals. Okay, so moving on to another kind of class of models. So Mark-recapture, I'll use this kind of hypothetical example to kind of explain the concept. So in this bag we've got some unknown number of marbles. And so what we're going to do is we're going to pull marbles out one at a time and we're going to mark them and put them back. So pretty soon we're going to have a bag that has some mixture of unmarked marbles that we didn't pick and marked marbles that we did pick and mark, right? So let's say we marked, for this example, let's say we marked 50 of them. Now we're going to resample the population of marbles in this bag, so we can reach in and pull a marble out and look at it. We can either set it aside or we can put it back. There's two different ways to do that. So we're going to resample it and we're going to make note of whether the marble we picked was marked or wasn't marked, right? We're also going to invoke the assumption that the fact that we marked the marbles doesn't make them any more likely to be chosen out of the bag, right? So marked marbles and unmarked marbles have the same probability of being pulled out of the bag. So if we marked 50 marbles and put them in the bag, and then I had you draw 100 times a marble and put it back, and 98 of those times you pulled a marble out and it was marked. So we won't go into the math, but what would that sort of tell you? Well, you'd probably logically conclude that you marked most of the marbles in this bag, right? So I don't know how many's in the bag, but it's probably not a whole lot more than 50. Probably fairly close. If we'd done the same thing and you pulled out marbles and you picked one out of that whole time that was marked, what would you think? Comparatively, you would say, well, there's probably a lot of marbles in that bag because we weren't very likely to pull one of these 50 marked ones. So that's kind of the concept. And then there's math that allows you to actually generate sort of specific estimates. So we basically are applying the detection rate for the marked objects, or marbles in this case, to the unmarked objects to estimate the total number of objects in the bag. So we can do this with animals. We can do it with fish. We can do it with mammals. We can do it with birds. We can do it with reptiles. So historically, those methods were generated around the concept where you went out and you trapped like small mammals, you marked some, maybe you toe clipped them, and then you had a resampling time where you came back and trapped the area again, and you physically captured the animals and you examined them for a mark. So what we've done with large ungulate surveys is we've sort of innovated that so that the recapture part of it is not really going to be a recapture. It's going to be a resighting. So we're going to mark some animals, and then we're going to try to resample the population, and what we're going to do is rather than catch them, we're going to see them, and we're going to note whether we saw a marked one or an unmarked one each time. Right? So there's your marked elk in the middle of a whole bunch of unmarked elk. In these kind of models, there's a few pretty common essential assumptions. One is that you assume that the animals that you've marked don't lose their marks, right? So it's fairly critical that you have a pretty good handle on how many marks are available to be sampled. If animals are losing their marks at some unknown rate, then your confidence in knowing how many animals you have that are marked goes down. Similarly, marked animals and unmarked animals are you never confuse the two. You never see a marked animal and say it's unmarked. You never see an unmarked animal and say it's marked, right? So the marks have to be sort of detectable enough that that's not going to happen. Early on, most of the models assumed that there was closure between marking and resighting, so that the number of objects or the number of animals when you sample the first time is the same as when you go back and do your resampling. So we've been able to lessen that assumption a little bit, and we'll talk about that on the next slide. But those are called closed population models, closed Mark-resight models, Mark-recapture models. And also among the assumptions with most early models was that all these animals have equal detectability. So you're just as likely to resample any marked animal, and marked animals have exactly the same sighting probabilities or detection probabilities as the unmarked. We know that that's really not true, right? I mean, the whole concept of the Sightability model thing was that you're more likely to see animals in big groups, you're more likely to see them when they're in the open. So we really know that that's not an assumption that's reasonable, but it was an assumption that was required for a lot of early models. And then you basically assume that the marked animals are a random sample, a representative sample of the unmarked animals, and although you want the markers to be detectable enough that you never make this mistake, or this mistake, you don't want them to be so conspicuous that they're the reason you detect the animal, right? So if you've got that changes the detection probability. So unmarked animals at that point are much more likely to be seen or detected or captured or whatever than unmarked animals, and that's problematic as well. So through time, people have worked a lot on these kinds of models, and we've generated some improvements. So one is we now have some models that people have generated where the closure assumption can be relaxed, and usually the way that works is that there are multiple occasion approaches. So basically, you don't just go out and do a single recapture or resighting survey. You do multiple recapture resighting surveys, and that allows you, to estimate the detectability of individual animals. Same thing here. These kind of models have gone a couple of different directions. Most of them have gone into a family of statistical approaches known as maximum likelihood, MLE. But there's also approaches that are based on a kind of a different statistical concept called bootstrapping. MLEs sounds like a big word, but you're actually familiar with some maximum likelihood estimators. So the mean, the sample mean is the maximum likelihood estimator for a population mean. So here's a variety of Mark-resight models. So one of the first one that was in use was called the Lincoln-Peterson Index. It required both geographic and demographic closure, meaning animals couldn't come and go between marking and surveying, and they couldn't die or be born. So you couldn't have changes in abundance because of that. Individual heterogeneity, so detectability among individuals was assumed to always be the same, and in those models, when you recaptured them or resighted them, you didn't care if you knew who the individual was. You just cared that you knew that they were marked. So that's called a batch mark model. So some models eventually, especially the ones that allow you to tolerate different sighting probabilities across individuals, they generally require that when you detect a marked individual that you actually are able to identify it. So that allows you to create an encounter history for each individual animal, and that allows you to estimate sort of the variation in detection probabilities. And so the model so there's been a series of these that have been generated, they've all been published in the literature, and the one that's a fairly recent addition, which is really pretty darn useful. It's a really good fit for Mark-resight kinds of things, is this LNME, which is basically the logit normal mixed effects model. It was developed by a guy named Brett McClintock, who's now at the Marine Mammal Lab in Seattle. He was a PhD student at Colorado State when he did this work. But he generated this brand new likelihood function that invoked a model that had a lot of desirable attributes. It tolerated heterogeneity and detection rates. It was very flexible, it was very robust, and so we've been actually using them. And lots of published literature on these kinds of things. So we have been using this approach with elk in particular in a couple of different places in Washington. And so again, here's the group of elk, and then here is one elk that's got a collar on it. So we can use the telemetry system on the helicopter to tell us, well, which collared animal is this? We make note of that in the data that we collect. Again, to deal with the heterogeneity thing, we need more than one survey, so we were doing more than one survey each year, each sampling season. Allowed us to assume closure only for the surveys that were done, the multiple surveys done within a year, but we didn't have to assume closure between this year's survey and next year's survey. Got the individual IDs, and because of that we can deal with this heterogeneity. And then we used a flight protocol that was pretty similar to what we did for those sighting trials for a sightability model. So we fly out there, we have some flight protocol, speed, altitude, and we collect data for all these groups, and we have, at the end, we have a a tally of unmarked animals we saw, we have a tally of marked animals we saw, and their identities. In this particular project, we had, in the helicopter, we actually had a GIS system running in the helicopter with an orthophoto that was sort of our background layer, and we could actually see where we'd been, where we hadn't been. We could see where the clear cuts were. We could see where all the kind of environments were that we wanted to sample in our survey, and that was really helpful. So then we flew a really large landscape where we did this. We flew all the habitats that had some sort of real non-zero chance of us detecting animals. We did it at the end of the winter, early in the spring, when we believe that animals are most likely to be in the open as the green up sort of starts. Took about 35 hours of flight time for every one of these individual surveys, and so again, at least 2 a year. And the data were collected by 3 observers and a pilot in a helicopter. And so these are Mark-resight estimates. This is for cows, so across years from 2005 to 2012. This was a population that was growing and was recovering from a sort of a historic low point in abundance. We had done some things to increase abundance, and so we were trying to track that with the Mark-resight approach, and that is the data that we got. This is for just the Branch Island bulls during that same effort and that same time frame, and you can see the estimates of bull abundance. Point estimates started around 90, and by the time we were done, they were up close to 300. So one of the things, and so what we did in several projects is we actually did the Sightability model development at the same time that we did Mark-resight. So we were flying surveys and we were collecting data that we were then using 2 completely different approaches to estimate abundance. And what we found was that the Mark-resight approach was clearly superior for accounting for animals that had very low sighting probabilities. The Sightability model had trouble with that, and it's basically because of the way it works. And so animals that were in really low sighting probability contexts environmentally, we didn't account for them as well with the Sightability model, which basically means that we always underestimated with Sightability model relative to Mark-resight. Our Sightability model estimates were always lower. But one of the things that was kind of encouraging was that when we took a bunch of those survey estimates from the same surveys and compared them from the 2 approaches, we found that they had a really, really high correlation. So what that suggests to us is that although Sightability modeling was not giving us unbiased estimates of true abundance, it was apparently tracking trend quite well. We were getting the same trend indication from those underestimates as we were getting from the better estimates of Mark-resight. Sightability modeling applications are way more practical because you don't have to have marked animals forever. With this approach that we were using, you could only do it if you have radio-collared animals. So if you're going to do that as your monitoring scheme, you got to have marked animals all the time. So distance sampling is that third class. It's a fairly intuitive concept. It's not one we've done a lot with for these animals, but we are actually currently starting to explore it a little bit. So there's some elk in a clear cut near Mount St. Helens. So the idea is it's fairly intuitive that when you're out trying to detect objects, whether they be elk in the environment or whether they be chunks of 2x4 laid out in a field, I mean, you can do this experimentally. I've done this in a class before where we used pieces of wood to do it. But basically, the assumption is that the objects, you have a higher chance of seeing objects that are close to the line you're walking, right? So things that as objects get further out, you have a lower chance of seeing them. That seems reasonable, doesn't it? And so basically what distance sampling is about collecting data and building this function that actually predicts what that shape of that curve looks like. So when things are very close to a transect line, you have very high chance of seeing them. But at some point, begin to decline, and at some point you become very unlikely to see an object simply because it's so far away from where you're at. It's smaller, it's further away. The fundamental assumptions is that on distance sampling are that you have perfect detection at distance equals 0, meaning you don't miss any animals that are on your line. It's not quite as hard an assumption as it used to be, but no movement prior to detection. So when you detect an animal and measure that distance, you're assuming that it didn't start on the line and you didn't detect until it was 50 meters off the line. Distances are measured without error. That seems pretty obvious. And then the transects are placed randomly relative to the distribution of objects. So the objects don't have to be randomly distributed, but the location of your sampling convention, your transect line, has to be located randomly relative to the objects. Okay, so that's an assumption. So the way you do this in an aircraft is you fly, and there's a couple different ways to do the distance thing, but essentially you're flying along, you're detecting objects like elephants, which we don't do that in Washington. And so then you're basically interested in the distance between the line you're flying and the animal's perpendicular distance to that line. So there's ways to do that. It seems a little, it is more challenging than doing it from the ground, but you can do it. Again, lots of literature sort of documenting applications of this approach. And so where we're exploring distance sampling is doing it with moose in northeast Washington. We just started a project this last winter, and we have a growing population of moose in northeast Washington. Most people know we have moose in Washington, right? They're actually becoming fairly numerous. They make regular appearances in the city limits of Spokane every year. We actually have, during the summer, we probably have our folks in our Spokane office probably deal with at least 1 or 2 moose calls a week. So again, here's the concept. We're flying some line in a helicopter, we're detecting moose, and we're interested in what's this distance. And so the data that we collect is a series of observations of moose. Some of them are close, some of them are further away, and the goal is that we build this function that predicts what that fall off is. So we just started this project. We started it by going out and radio collaring some moose. Collared 30 moose in a week just last December. We're going to collar another 30 this December. And the collaring was actually to enable us to do both the distance sampling and the Mark-resight thing consecutively, so that we could sort of compare those results and see. Moose, why haven't we done more distance sampling? Distance sampling is a really good technique. It's really robust. So a lot of the things that you might assume are problematic with things like the animals are not all equally detectable. Some are under a bush, some are out in the open. Distance sampling is really quite robust to those sorts of issues, and so it's a really nice technique. It's got a long history. The primary think tank for distance sampling is a group of ecologists at St Andrews University in Scotland. They've written pretty much the books, developed all the software. The reason we think moose is a really good candidate for a distance sampling approach, and why some other ones that we don't deal with. So Mount St. Helens was an environment where we had lots of elk when we started that project that I showed you the other estimates from. So as you can imagine, in a helicopter, dealing with seeing animals out of the helicopter, and while you're circling them to find out how many calves are in that group and how many bulls are in that group, somebody sees, oh, there's some other elk over there. They're really not on the line we were flying anymore. We saw them because we stopped to fly around these guys. And so pretty soon it would get really, really crazy in terms of, pretty soon you'd say, I don't have any confidence at all that the distance thing is really working, or that we're seeing these animals because of their distance from the line we originally were on. Moose are a pretty good candidate because they're not as abundant. They tend to occur in the environment as not always single individuals, but usually group sizes are pretty small. And so they seem like a pretty good fit for us to try distance sampling. We think it seems like a much more doable equation, not likely to have as much sort of noise associated with just the way the survey's done and things like that. So that's kind of what we're doing with that. So here's just a kind of a summary of how we're applying some of these methods across the state of Washington. So the Sightability modeling approach, we're actually using that to monitor an elk population in the Blue Mountains in southeast Washington, the Yakima elk herd, the Cloquin elk herd, the Hanford elk herd, the Mount St. Helens elk herd, what we call the South Rainier elk herd, some of those elk you see in Packwood. In fact, you might get close to running one over in Packwood one time. Columbia Basin mule deer was that project I talked about, and then Blue Mountains mule deer as well. Mark-resight we don't do as in as many places. You sort of need the right set of conditions, and so again, you're typically you're in this realm of having to have marked animals all the time for the model that we've been using. So we're doing that with this small northwest Washington elk population in the Nooksack. We did it for Mount St. Helens elk, but it's not the long-term plan for Mount St. Helens elk. This is the long-term plan right now for Mount St. Helens elk. We've done it for elk that occupy the alpine zone in Mount Rainier during the summer and fall. And we used it in a few places on the Olympic Peninsula for elk. Distance sampling, which is sort of the only place that we're really dealing with that right at the moment, is with moose in northeast Washington. So the tough nuts to crack in this are these situations with very low detection rate context. So this is a picture when we were doing our work in Nooksack. So we did our survey, we counted for all the marked animals that we saw, and we started doing the phase where we tracked down the missing ones. Took us to this really dense stand of trees. We knew we were right over the top of this radio transmitter, couldn't see anything, we just kind of kept flying this little circle. And eventually this head slid out from under the trees and kind of looked up at us. And there she was, and you can see her collar there. But those are the kind of situations that if you have that, if that's a common scenario where you're working, that's a problem. It becomes a very difficult problem to overcome. So west-side black-tailed deer are kind of one of these low detection rate contexts. Northeast Washington deer and elk up in the Selkirks and some of that country, the Kettle River country. Eastern Washington white-tailed deer, they act differently, they behave differently than mule deer, and it makes them kind of difficult to deal with in terms of sampling. Ideally, we'd like estimates. So we have these models that help us account for missed animals, right? So the final estimate is a mixture of animals we saw and counted, and some animals that the model told us we probably missed based on something. What we like is we'd like estimates at the end of the day where most of the individuals in the final estimate came from animals we saw. That's the ideal scenario, right? There's less uncertainty with animals you saw. Having an estimate at the end of the day where most of the animals in your estimate came from the model predicting they were there but you didn't see them, that's not as desirable, and that those tend to be less precise estimates anyway. So sometimes for situations like that, the best approach isn't sort of this direct enumeration thing. Sometimes it's what we call a population process model. So it's possible, theoretically, to estimate something like abundance or age structure by estimating those quantities based on something else that you know that's more easily measured. So you can take demographics, things like survival, reproductive output, and it's possible, in theory, to use those to predict things that are not so tractable. Complexities for doing this range from spreadsheet models. I've built a number of spreadsheet models that basically are sort of an abstraction of a population, right? So you've got individuals, and you apply some survival rate to them, and you apply some reproductive rate to them, and you can track that through time under those sort of constraints. Really pretty easy to do in a spreadsheet, but you can also do it in really complex ways. I was going to put a likelihood up here, but I figured that would just scare everybody. But there's some really heavy lifting mathematics to some of these approaches. But unbiased and precise estimates of abundance. It'd be nice if it was magic, but it's not. So you really can't derive good estimates from no data or crappy data, right? So you have to have, yeah, you can try, but good luck. Some of these approaches are really data hungry. You have to have some piece of it that you have fair confidence in, that you know with some reasonable confidence. But again, some of these are really data hungry. You need individual ages. You need good survival estimates. It's better if you actually have even some estimate of abundance for a subset of your population. You know, those can be used, but and this is an area where a lot of work's actually ongoing now. There's the literature is pretty regularly being updated with new approaches. Some of it is, if you look at some of these methods, it seems pretty far-fetched. I mean, if you look at sort of what the approach is and what the assumptions are required, it would seem pretty wild to the average person. But anyway, it is an area that there's a lot of really smart people working on these kinds of solutions. And for some of those situations where we have not had much luck, sort of with direct enumeration, we're using traditional approaches. This may be the only thing that we can do. And so that's all I have. So I'd be happy to try and answer any questions if you have any.